Assalamualaikum warahmatullahi wabarakatuh and greetings to the Secretariat of International Summit. My name is Putri Nur Aisyah binti Ahmad Hashimuddin Salih, and today I will present to you about my research study, Phytolith extraction and cataloging of Dipterocarpaceae, upper hill species, and site-specific Palaeoecology biomarker. Nowadays, climate change has become a global issue. Shifting temperatures due to climate change are driving changes in global biodiversity as species are struggling to adapt to the rising temperature globally. So in order for us to document these environmental changes, a diverse range of fossil types have been used, such as pollen grain, ice cores and glaciers, and also phytolith. Pollen grain is said to be a useful and powerful biomarker. This is because they are protected by their durable outer layer, and it can survive and become embedded in sediment layers at the bottom of the water bodies. Pollen records provide a window into the past, allowing us to understand natural climate variability and the significance of human activities on Earth ecosystem. While pollen grain have said to be a powerful biomarker, they also have their own limitations. When pollen grain are dispersed by the wind, it makes it difficult to determine the source region, hindering precise reconstruction of past vegetation. Meanwhile, on the other hand, phytolith is much more durable and have a clearer evidence in tropical environments. It is highly resistant to decomposition and usually preserved in soil or artifacts, making it as a good biomarker for climate change. Phytolith analysis has been demonstrated to be an effective method for reconstructing past climates. This is because the color of the phytolith can tell us if the organic material around it has been burned by fire and much more. So what exactly is a phytolith? Phytolith is a microscopic silica substance that we can find inside the various cells of the plant. It is very tiny in sizes and we cannot see them using a naked eye. Phytolith function as structural support and defense mechanism for the plant. Typically, phytolith color is white or colorless and they owe the appearance of their silica composition. So due to phytolith silica composition, phytolith are resistant to decay and this makes phytolith as a powerful and good biomarker in order for us to record the climate change. So the problem I have in my research study today is we have a lack of past reference and study on phytolith, especially on family Dipterocarpaceae. Family Dipterocarpaceae is the largest woody family that we can find in Southeast Asia region. So my objective in my studies today is to extract the phytolith from family Dipterocarpaceae, specifically from genus Dipterocarpus, and to cataloging all the phytolith according to the International Code for Phytolith Nomenclature 1 and 2. For materials and method, firstly is the sample collection. I have a total of 15 samples from 6 different species that were provided by my supervisor, Dr. Fong Sui. All the samples were collected from 3 different herbariums. They are located in Sandakan Herbarium, Sabah Herbarium, and Berau Herbarium in Jakarta, Indonesia. Next, for phytolith extraction, I use a chemical oxidation method in order to break down the organic compound into gases and silica intact. So firstly, I weigh the leaf sample of approximately 0.2 gram. And next, for chemical oxidation, the leaf sample were digested in nitric acid and potassium chlorate at 90 degrees water bath inside a running fume hood.
The next one is centrifugation. The samples were centrifuged and rinsed multiple times with distilled water in order for us to cleanse the acidic substance from the samples. The next step is the final residue was left to evaporate at room temperature until it is fully dry.
When the final residue of phytolith are fully dry, it is ready to be observed, so we must prepare a slide. The slides were labeled, cleaned, and smeared with a little bit of phytolith samples, and we top it with a balsamic oil, and then we cover the slide with a cover slip. Next, we take the slide for microscope observation, and we examine it under a microscope in order for us to observe the phytolith shape and morphology. So this is the overview of the result that we get from the phytolith observation. We observe that the major phytolith have a consistent shape and surface ornamentation across the 15 samples and 6 different species, which is spheroidal plicate. So how do we determine this surface ornamentation? We use ICPN 1 and 2 in order for us to determine their shape and their surface ornamentation. And we also found other minor shapes, and that shapes are really varied. This is the first three species of Dipterocarpaceae species. So we found that the major forms of Phytolith is consistent in terms of their shapes and surface ornamentation. The first species is Dipterocarpaceae caulifera, where we have only one replicate or one samples for this species. And next is Dipterocarpaceae fusiformis, where we have two samples. And next is Dipterocarpaceae crinitus, where we have four samples. And this is the next three species. Next is Dipterocarpaceae gracilis, where we have two samples. Dipterocarpaceae geniculetus with three samples, and lastly is Dipterocarpaceae grandiflorus with three samples. Most spheroidal phytolith reflect the rounded shape of mesophyll cells in which they are formed. Plant cells have different structures, some with smooth walls and others with textured or patterned walls. So when silica deposit within these cells to form phytolith, the resulting surface reflects the original cell wall structure. So besides the major forms of phytolith that we found in 15 samples, we also found other forms of minor phytolith such as acicular entire, trachary annulate, rectangular plicate, polygonal fuvose, and much more. When we have already determined the shape of the major phytoliths, we take 20 best shaped major phytoliths of each of the 15 samples for us to do a morphometric analysis where we measure the length and the width and we take the average of each length and width. Next, we will use SPSS software to do an analytical test. So first analytical test is the normality test. So how do we know which normality test are we using? Because our sample size is 20, where we take 20 best shape of the major phytoliths. So we will use Shapiro-Wilk normality test. This is because the sample size for Shapiro-Wilk must be less than 50. Based on the result of the normality test, we can determine which statistical test we can do next. So if the p-value of the normality test is more than 0.05, which means the data follow a normal distribution, so we can proceed with parametric test. Meanwhile, if the p-value for normality test is less than 0.05, the data does not follow the normal distribution, so we must proceed with non-parametric test. For parametric test, we will use one-way ANOVA. Meanwhile, for non-parametric test, we will use the Kruskal-Wallis test.
For the statistical test, there are two parts, which is the first one is between the same species, and the second one is statistical test between the 6 different species. For first part, first species is Dipterocarpaceae caudifera. However, we only have one replicate and one sample for this species, so there are no analytical comparison that can be done between the sample of this species. The second species is Dipterocarpaceae crinitus. We have 4 samples for this species, which is sample 2, 3, 4, and 5. When we do the normality test, it is reveals that it met the assumption of parametric. So we can do a one-way ANOVA test. One-way ANOVA reveals that there are a significant difference in terms of length and width of the major phytolith for this species. So we do a post hoc test for us to find out where exactly the difference lies between between the samples. So we found out that samples 2, 3, and 5 are significantly different from one another. The next one is Dipterocarpaceae fusiformis. We have 2 samples, 6 and 7. The normality test reveals that it met the assumption, so we can do a one-way ANOVA. One-way ANOVA reveals that there are no significant difference in these 2 samples. The next species is Dipterocarpaceae geniculetus. We have 3 samples, 8, 9, and 10. It met the assumption of parametric test, and one-way ANOVA tells us that there are a significant difference. Post-hoc test reveals that sample 9 and 10 are significantly different. For Dipterocarpaceae gracilis, we have 2 samples. It met the assumption of parametric, and one-way ANOVA tells us that there are no significant difference between these 2 samples. The last one is Dipterocarpaceae grandiflorus. We have sample 13, 14, and 15. Normality test tells us that it met the assumption, and there are a significant difference in terms of length and width. Post-hoc test reveals that all the samples are different from each other. The next one is statistical test between different species, which we do a statistical test between the length of 6 different species and the width of 6 different species of the major phytolith. So for the length, the normality test, it met the assumption of parametric, so we can do a one-way ANOVA. And one-way ANOVA reveals that there are no significant difference in terms of length between the major phytoliths for all the species. Meanwhile, for wheat, it does not meet the assumption of parametric, so we must do a non-parametric test, which is Kruskal-Wallis. Kruskal-Wallis reveals that there is a significant difference. So, in order for us to find out which species have different in width for their major phytolith, we do a post-hoc test. So, post-hoc test reveals that Dipterocarpaceae fusiformis is different from Crinitus, Geniculetus, and Graniflorus, and Graniflorus is different with Fusiformis and Gracile. For discussion part, we will discuss all the 6 species differently. Firstly is Dipterocarpaceae caudifera. It was taken from Herbarium Sandakan in 1961. This species is native in Borneo. And in these studies, we only have 1 sample, so we cannot do any statistical test for this species. And there are also no studies or reference on where is the habitat actually grown. The next one is Dipterocarpaceae crinitus. We have 4 samples for this species, and the 4 samples are collected from different herbarium, which are at Sabah and Berau. So the sizes of phytoliths show variation in terms of length and width across these 4 samples. And this species can be found in both lowland Dipterocarp forest and hill Dipterocarp forest. So these 2 regions actually have a different soil condition. So if the 4 samples were collected from different regions, this will explain why we can see that the variation of the size of phytoliths across the samples, even though they are from the same species. For Dipterocarpaceae fusiformis, we have 4 samples, and both are collected from the Sandakan herbarium. And in this study, we can observe that there are no significant differences shown in phytolith sizes in terms of length and width across the same species. For Dipterocarpaceae geniculetus, we have 3 samples that are collected from Sandakan and Sabah herbarium. From the statistical analysis, we found out that 1 sample has a bigger phytolith size than the other 2. Next is Dipterocarpaceae gracilis. We have 2 samples that are collected from the same herbarium at Sandakan. The length and width show no significant differences. These species can be found at upper Dipterocarp forest and hill Dipterocarp forest. So these 2 regions actually have a similar ecological condition. So if the samples are collected from these 2 different regions, that actually explains why we cannot see any significant difference in terms of the length and width. It's because they grow in a similar ecological condition. The last one is Dipterocarpaceae grandiflorus. There are 3 samples that collected from the same herbarium at Sandakan, and we can see such a huge difference in terms of phytolith sizes. These species can be found at 3 different regions: hill Dipterocarp forest, lowland Dipterocarp forest, and coastal variation. So these 3 regions show a different variation in elevation and environmental condition. So this explains why we can see such a huge difference in terms of phytolith sizes, even though they come from the same species. Even though my research studies today a lack of reference and information on where exactly and specifically my species grow, however, we do have previous studies that explain and prove that the phytolith sizes actually can be influenced by the environmental condition around them. For example, there are studies about the sizes of bollivon phytolith from a white rice, where these plants actually prefer water and wet habitats rather than the relatively dry condition. This is because they found out that the phytoliths from the water and wet habitats are mostly larger than those of white rice under the relatively dry conditions. Next examples show that the phytolith sizes can be influenced by the soil type. So if the plant grow in a preferable conditions and a preferable environmental conditions, the phytolith sizes will grow much more larger rather than the plants that grow in the soil that they are not preferred. So these references and these case studies show us that the phytolith sizes can be influenced by the environmental conditions. In conclusion, some species show significant differences in phytolith sizes, such as Dipterocarpaceae crenatus and Dipterocarpaceae geniculetus. Across the different species, we can see that there are no clear differences in length across the different species. However, the width varied, and the phytolith shapes were similar across the species within the same family of Dipterocarpaceae, which is spheroidal decade. In this research study, I believe the environmental condition might influence the phytolith sizes, even though they are from the same species or same families. However, we do not have enough past reference and past studies to support this. So, in future studies, I suggest that we collect more samples and more environmental data, such as soil type and precipitation and environmental condition, in order for us to make phytolith as a remarkable and as a useful and powerful biomarkers for us to measure the climate change globally.